Our guest speaker, Dr. Erika Calvo-Ochoa, is a molecular neuroscientist. She earned a bachelor's degree in biomedical research and a doctorate at la Universidad Nacional Autónoma de México, affectionately and pragmatically referred to as UNAM. She received a fellowship from the National Science Foundation to allow her to pursue her research in Kalamazoo at Western Michigan University. She liked Michigan so much that she joined the faculty at Hope College in the summer of 2020, in the middle of a pandemic. She spoke to HAS members during the summer of 2022. Her presentation then focused on the major features and mechanisms of the receptors for temperature and touch. The researchers who had uncovered those mechanisms had been awarded the Nobel Prize for Physiology or Medicine just a few months earlier. She explained how these signals are sensed by the brain, and that's right up her alley. Her research focuses on studying molecular and cellular markers of neurodegeneration, loss of brain plasticity, and inflammation following brain damage and injury. She has explored various animal models over the years. She currently works with the zebrafish. Why the zebrafish? It turns out that the zebrafish has an olfactory system which is very similar to that of mammals, and their brain exhibits a remarkable degree of plasticity. It can also regenerate throughout life. Using the zebrafish brain as a model, she explores both neurodegeneration and regeneration mechanisms following brain injury and disease. Her latest research paper was published in May in the International Journal for Molecular Sciences. It's titled Olfactory Dysfunction in a Novel Model of Premortal Parkinson's Disease in Adult Zebrafish. I leave you with the following intriguing question. How are zebrafish and the United States Postal Service related? For the answer to that question and many more, I turn it over to Dr. Erika Calvo-Ochoa.
Thank you, Luis. Can everyone hear me well? Yeah, David, over there. Wonderful. Thank you for this wonderful introduction. You pretty much did the introduction for half of the presentation so far. So I hope that I can live up to that expectation. And yes, I came in 2022 to talk about the Nobel Prize of Medicine in 2021. This is nowhere near Nobel Prize science, but it's a science that we do here at Hope College with undergrad students. So I'm really proud to present to you what these wonderful future scientists and doctors do in our lab. As Luis mentioned, we study zebrafish and we particularly focus on the olfactory system. And I hope that I can convince you that olfaction is the best sense that there is. We usually don't hear that. It's kind of like, no. Vision is the very best. Maybe audition is the second best. And olfaction, we kind of think that it's not as important for us as it is for other animals. So the sense of olfaction, of course, allows us to enjoy multiple things around us. Hopefully, if you guys like to grill during the summer, right? And you can go to your friend's house and you immediately know that some sizzling party is happening, right? Because of those grilling aromas. And most animals use their sense of olfaction, like us, to experience the world. And something important about olfaction is that it drives some of our emotional responses, right? Think about an odor that really evokes something really nice, like a food you like, or maybe the smell of a loved one, and maybe other smells that are not as good. The emotional response is not going to be as positive, right? So the sense of olfaction has been historically embraced and celebrated, and we know this because there's many depictions of perfume and fragrances from the older times, mostly in Eastern Roman and Greek cultures where perfumes were incredibly important. But somehow, throughout history, more of the Western civilizations in Europe started losing that connection with smelling well. Okay, so one of the earliest depictions in the modern Western civilization is this. Like, does this ring a bell for anyone? What is that? Someone said yes over there. This one over here. That's a plague doctor, right? It's a plague doctor where odors started to gain an important role because, I'm sorry. They started putting these herbs, right? Because they thought that some of the odors that were coming from people with the bubonic plague were actually bad. But before that, the sense of good smells weren't as important as for other civilizations. Okay, so about 150 years ago is when these philosophers and scientists started discarding these the sense of smell, right? So for example, Descartes and Broca started pronouncing that the olfactory bulb, which is in part of the olfactory system that we will chat about in a few seconds, it's found in other animals and it's very large, but in humans it's quite tiny. So maybe it is not as important for our species as it's for other species, right? Like dogs. We know dogs are really, really good at smelling. And we also have this Western philosophy of Descartes that pronounced that visual input was the most important for us. And because humans are logical beings and olfaction seemed to be driving some of those primal responses in other lower animals, we might not be that olfactory driven, right? This was 150 years ago. And this was kind of a dogma that some neuroscientists continue to have until very recently. Okay, so the field of olfaction has been actually very, very understudied. Again, why would we study olfaction if it's not that important for us humans? So the mechanisms behind odorant recognition were only discovered in 1991. And when I say this, I was not only alive in 91, I do remember 91, but can you imagine my students who were born in the 2000s? To them, that sounds like 300 years ago. It's like so last century. But for us, 1991, it was basically yesterday, right? So this is very recent. And the person who discovered this was awarded the Nobel Prize in Medicine in the early 2000s. This is very recent. And this is one of our 5 senses, and it was only discovered in the 90s. So from the 90s to now, we can know now that olfaction is actually really important for humans, and we're actually quite good at it, even if we don't understand that fully. It is thought that humans can discriminate more than 1 trillion olfactory stimuli, trillion, okay? So we might not have as sensitive of a nose as our furry companions, but we are really good at discriminating odors, okay? We can tell a pie, whether it's an apple pie, whether it has cinnamon on it, whether there's a lot of butter in the crust, whether there's some chocolate, maybe. We can discriminate these smells really, really well, okay? And I actually like to show this example, which I find it to be both hilarious and illuminating. This is a group of scientists who study human olfaction. They don't work with other animals. They only work with humans. And I actually met this investigator last year at a conference. And I was like, I always talk about your experiment of human tracking. How do you do that? And I'm going to tell you a little bit more about it. So you have probably seen or are familiar with how tracking dogs can follow a prey. right? They will sniff around, and they will probably follow this very winding shape until they can track their prey, right? Dogs are really, really good at tracking. But what about humans? Well, we are actually quite good at tracking. We don't have to do it often, but when we're asked to do it, it turns out that we could, actually. So in these experiments, all the subjects were wearing a mask that was outfitted so the investigators could report the amount of sniffs and the intensity of the sniffs. And these participants were blindfolded. So they were not using any visual cues of any sort. And if you can see this, humans actually are pretty good. Again, we don't have to do it often, but maybe, I don't know, some millennia ago, we needed to track something. So we were actually quite good at it. And what was used for tracking this is chocolate essential oil. Okay, chocolate will motivate most of us to track that scent. So can you imagine? So I asked the investigator behind this, like, how do you even come with ideas like that? And he told me, we were having a lab picnic and one of my students dared the other, like, I dare you that you are not gonna be able to track where the hot dogs are using your smell alone. And the guy was like, is that a bit? And meanwhile, the head of the lab was like, this is a paper, this is a study, and this is it in a very prominent neuroscience journal. So yeah, you know, I'm just hoping to get some of those insights, maybe like having coffee with my students, and maybe someone will come up with a genius idea so far. But yeah, this is fantastic. We are able to smell just right. Okay. And something important about olfaction, again, we talked about survival and emotions and all of that, is that we share some similarities with how we use olfaction with other animals. For example, and these are all studies in humans, just to show you an example of how good we are at smelling things, right? So we possess the ability to discriminate food fat content just by smelling it. For example, like a crust, like a pie crust, we can tell sometimes which one is fattier than the other. Sometimes if you're really, really good, and if you have a very acute sense of smell, you can tell whether it's butter or something else, right? We are able to discriminate fatty foods from non-fatty foods using the nose alone. Mating, okay, we think that maybe we chose our partner just based on their intellectual abilities and, you know, our shared interests. But it turns out that body odor is important for finding a partner. Okay, so and I tell this to my students who are in the dating scene, maybe we're not anymore. Like, you know, when you go out, you better smell good because people is actually smelling you, unconsciously deciding if you're gonna be a good partner or not. So we're actually to even understand other people's emotional state through smell. Like, for example, tears have important chemicals that we can detect using olfaction to gauge emotional state in others. We obviously don't do this consciously, right? Like, oh, my partner's tears smell like they're in distress for this or that, but we can actually get those emotional cues using olfaction. How wonderful is that? And we can also use this for alarm responses, okay? So we can tell, of course, when some foods are spoiled, but it turns out that we can even smell sometimes disease in others. I read this fascinating article in the New York Times about this woman who, I believe she's in the UK. She can smell Parkinson's disease in other people. there's something quite unique. There's something in Parkinson's disease bodies that she can identify. And she's like, I want scientists to use me to figure out why I can't smell Parkinson's disease. And people sometimes discredit her, like, well, not quite. But there are dogs that can smell, for example, for cancer and other diseases. So we might be able to detect some of these cues as well. So because olfaction is so important, one of the questions that my lab asks is if olfactory function can be lost, and what are some of the consequences of losing this critical sense of smells, okay? So just a very quick recap of how we can perceive odors. So every chemical that has a smell that we can detect as a smell, we're gonna call odorants. Not every chemical we can detect. And I work with fish. So not every chemical that the fish can detect, we can detect. So an odorant will be a chemical that will bring a particular scent. So all these odorants, in our case, are flowing in the air and will hit the very top of our nose. This is called the olfactory epithelium. And I'm just going to start using, of course, these very scientific technical words. If you need me to stop or to go back or have any question, please raise your hand. I love to answer questions as we go, okay? So the olfactory epithelium is the epithelium in our nose that is exposed to the air, or in the case of zebrafish, to the water, and is connected directly to the brain, to a region called the olfactory bulb. So that is a part of the brain that actually processes smells. And they're both connected by this olfactory nerve, okay? And in the brain, we process what are we smelling, how are we going to respond to these smells? Is this gonna elicit a behavioral response or not? And something important is that olfaction is necessary for taste perception. I imagine all of you have had a nasty cold with a lot of mucus when you can't taste anything, right? You can't perceive any odors and you can't taste, and it's quite annoying to be honest. Okay? So again, can we lose our sense of smell? And we actually can lose our sense of smell. And olfactory dysfunction is actually quite common on people who are aging or losing youth or just becoming seasoned in life. So just aging itself, just like with hearing, a lot of older adults start losing some of that important hearing. Olfactory starts to be decreased as well, just with age. But there are there are conditions like traumatic brain injury, ischemic stroke, Parkinson's disease, or Alzheimer's disease. All of these neurological conditions can be associated with olfactory loss. So up to 96% of people with Parkinson's disease will have a degree of olfactory loss. And from 85 to 90% of people with Alzheimer's disease will present olfactory dysfunction, which is quite a high number. Okay, so those are related. So as I told you before, the sense of the field of olfaction wasn't necessarily, and I'm going to use this very technical term, the hottest in neuroscience, right? We were kind of like, well, you can study olfaction, that's fine. But then 2020 came about, the pandemic. And then I remember this very vividly. In April or so, I started getting emails of excited scientists who were, of course, very worried about the pandemic, but they were excited because people were reporting loss of olfaction and taste. So they were like, I guess we are hot. It's the time that olfactory scientists became hot. So these are Google Trend searches. You can use this feature, Google Trend, and you can look for any word or any sets of words, and you can see how many searches that particular term has on Google. That is very useful for us to know trends. And this is the amount of searches for smell and taste loss starting in 2018. So basically no one cared. And look at this. This was early 2020. People started to lose their sense of smell with COVID and started Googling it, and people starting to get really interested in this. Did any of you lose the sense of smell or taste with a COVID infection? Yeah, I did. Did it come back? Not totally. Yeah, did it come back? No. Yeah. So a lot of people experience that. It didn't come back fully, or it came back, but weird. So a lot of people then started to realize that, boy, smelling and tasting is actually important for my life, right? So this is where we started to, this is where the general public started to realize how important olfaction is for health, for establishment of social connections, and why olfactory dysfunction is a serious public health concern that we should really, really address. Okay, so these are basically the 3 questions that we want to study in my lab. I hope that I sort of convince you that olfaction can be important, and then we can detect other types of chemical cues in the air. So our questions are, number 1, if the olfactory system is able to repair and regenerate after injury and disease, like COVID, ischemic stroke, brain trauma, all of those. If we can recover olfactory function after time, and if this is possible, what factors drive olfactory regeneration? Okay. And as Luis very kindly introduced this talk, we know that the nervous system of mammals, like you and me and rats and mice, which are the most widely used animals in neuroscience research, we're sadly not good at regenerating neurons. If we get a neurodegenerative disease, or if we get brain trauma, the recovery is not going to be complete, sadly. Is going to take a long time. So we are not the model to study if we want to study recovery. So we use this fantastic model, which is the zebrafish. So zebrafish are actually like 1.5 inches. They're quite tiny, so you're going to see them gigantic in your screen. They're actually quite tiny. But they're wonderful, wonderful models because they're sort of similar to humans. They share about 70% of their genome with humans. So that makes them close enough to us that we can use some of the same genes that they have to study the genes and their pathways. But they're also different enough that we can use them to study these regenerative processes. And as Luis mentioned before, he posed a question to all of you, right? Like, what is the connection between zebrafish and the postal system? Yes. So zebrafish has been immortalized in a stamp in a collection in 2023. It's called Left Magnified, and they're all wonderful and beautiful images that different scientists around the world have taken. They're just breathtaking. And zebrafish was immortalized there. Of course, I got as many as I could, and I will never send a letter to you using my precious zebrafish labs. But this is wonderful. So you didn't see that coming, right? Yep. Do you have any question over there? No, no questions so far? Okay. So we're going to get more technical, and I'm going to tell you a little bit more about the olfactory system of zebrafish itself. Just like in that slide where I showed you how it looks like in humans, in zebrafish is quite similar but simpler. So it's easier for us to manipulate and to study the system. It's much simpler. So you're gonna see a lot of images like this, where we have a black background and lots of pretty bright colors. I'm gonna explain to you how we do those in a second. But what you can see here is the olfactory epithelium that is exposed. It's out in the nose. And the olfactory bulb, which is a part of the brain. And these two are connected by the olfactory brain. In zebrafish, the olfactory epithelium is quite large and it's quite beautiful looking. We call it rosette because it does look like a flower. It's really, really beautiful. So you might hear me talking about olfactory epithelium or rosette. Just for simplicity, you're gonna see that I often will use OE as olfactory epithelium and OB as olfactory bulb, just for making it easier for me to have them on my slides. So if we were to look and magnify these rosette, these olfactory epithelium, we would find individual neurons lying there beautifully. These are called olfactory sensory neurons. And you know, a lot of things sometimes in science don't make a lot of sense, but when it comes to olfactory system, everything is called olfactory something, and it usually tells you what it is for. So an olfactory sensory neuron is a neuron that senses odorants, right? So these neurons are the ones that are exposed to the environment, and these are the ones that will receive all of these chemicals that we respond to, okay? And this is a diagram of the olfactory bulb. You're gonna see that quite often throughout the slide. Do we have any questions so far? No questions? All right. So how does this work? So as I told you, odorants are chemicals that we can smell that, elicit a response. So these odorants, whether in the air, in our case, or in the water, in the case of zebrafish, will bind to the epithelium and will activate different olfactory sensory neurons. When these neurons get activated, they have long axons that we call olfactory nerve, and these axons will fire a signal in the olfactory bulb, right? So we're talking neuron to neuron communications, synapses, all of that. All of this is sensory systems. So these odorants get processed, they get received in the olfactory sensory neurons in the epithelium, but they get processed in the olfactory bulb. So this communication is fundamental for us to be able to discern smells. And in the case of zebrafish, well, in our case as well, but particularly in the case of zebrafish, olfactory processing often leads to differences in behavior, okay? We're going to talk about behavior later. So we're lucky that zebrafish responds very strongly and very stereotypically to different odorants. For example, if we expose zebrafish to a chemical that smells like food, they respond just like if we smell something that smells like food. And if we didn't have lunch, didn't have time. So when I'm out of here and I smell something good, I'm gonna have a response, right? Zebrafish have the same response. If zebrafish smell a potential mate, if a male smells a female, and they're alone. We do this in the lab because we breed them, and they're alone and they have the right light conditions and the right temperature and the right environment, there will be love, okay? And this is just because of those chemicals that are secreted, pheromones and such, that will be secreted by the female. We also can test alarm responses. Fish can also respond very strongly if they smell something that is going to be harmful for them. And we can measure that, and we do that in my lab. Okay? So as we have mentioned before, one of the reasons why we study zebrafish is because they're very regenerative. Unlike humans, roughly speaking, humans do not generate new neurons throughout adulthood. we are born with the amount of neurons that we're going to have. We actually lose neurons when we're kids. We have more neurons than we need. And sadly, we do not have more. So this is why it's important to take care of our brains, right? And take care of those connections, because we cannot create more neurons. But zebrafish can, and they create new neurons throughout their lifetime. Actually, they have, and I'm not going to talk about this much, but I just wanted to show you that in the zebrafish brain, there are 17 places where new neurons are generated constantly. 17 places. We call these neurogenic niches. Neurogenic as if they generate neurons. This is a diagram of a zebrafish brain viewed from the side. You're going to see a lot of my results look like this. And at the very tip, we have the olfactory bulb right here. Oop, I'm sorry about that. So the olfactory bulb is neurogenic niche itself. It regenerates pretty fast. And this is the zebrafish rosette, or the olfactory epithelium. And all of these red dots are new cells generated. Again, incredibly regenerative, incredibly good at repairing. And this is the reason why we use these animals, okay? So for our first question, if the olfactory system can repair and regenerate. So I have 3 current ongoing projects in my lab. One of them deals with traumatic brain injury. Another one deals with, if not stroke, but low oxygen in the water. And the third one deals with Parkinson's disease. And this is a project that I'm going to tell you about today. It's our very last paper that Luis mentioned. And I feel like talking about Parkinson's disease is very interesting for people, because we probably all know someone who has Parkinson's disease. And if we do, I imagine that you maybe didn't know that this person very likely cannot smell well, right? Because some of the symptoms of Parkinson's disease are so severe. Sometimes olfactory loss, it's either not reported, or maybe it's not given as much consideration as other symptoms. But something that is incredibly interesting to me is that 96% of Parkinson's disease patients will present the olfactory loss years before the motor symptoms, right? So most individuals who get diagnosed with Parkinson's disease usually go to their doctor for motor symptoms. By then, it is likely that olfactory function has been decreased. So that's important. When I go to olfactory meetings, there's always groups of scientists who get together and ask, how can we spread the word to physicians? And I'm not a physician myself, so I don't have these answers. So then can be olfactory screenings when people maybe after a certain age get routinely screened. Actually screening olfaction is not very hard. There are like different tests where you are just asked, like, has anyone had any olfactory screening done before? You have? Can you share with us how this look like? Oh, yeah, so sorry. We need a microphone. I forgot. Sorry about that. Thanks.
The Parkinson Foundation put out some smell strips some time ago, and I just took that test, and they just had strips, and you identified them. I don't think they ever told me what I got right or wrong or anything like that.
Maybe you got such good olfaction that they didn't tell you, but they're pretty simple, right? They're like rose, cinnamon, spearmint. Yeah. So, and there are different types. Thank you so much for sharing with us. And there's one that you have to smell, I think 5 or 6. There's one that is more complex. You can smell up to 20 different things. And these are very simple. These are just You smell and you said, is this a rose? Is this clove? Things like that. So it's not very expensive. It's not very complicated. And it's something that physicians can do in their offices. I'm not exactly sure. We're probably not conveying these with enough to physicians, right? Because olfaction, who needs that? So it is important now, if you're curious, to perhaps ask your doctor, can I do one of these, and just check how olfaction is looking like. And something that I will say, I lost my olfaction due COVID, but it was immediate. Like one day I woke up, couldn't taste anything, went running to the spice jars, just like, okay, cinnamon, gone, clove, gone. I was like, okay, this is my life now. And no, being an olfactory scientist myself, I was like, oh my God, is it gonna come back? It came back fully, which I'm really grateful for. But olfactory loss during aging and other neurological conditions doesn't go like that. It's very, very slow. It's gradual. So I can imagine why some people don't even notice it anymore, right? Because it is so slow, it's so progressive that sometimes we don't know. Could I identify clove if I smell it? Could I identify spearmint? So I encourage you to try some of these, or ask your physician about this, and see if they were willing to share these olfactory strips with you. So yeah, coming back to Parkinson's disease and the project that I will talk to you about. So Parkinson's disease has many, many symptoms besides the most common and widely talked about, which are motor. There's a whole motor skill symptoms, but there's also non-motor symptoms, including the reduced sense of smell, okay? And again, smell is lost years before motor symptoms. So it's not that smell goes after everything goes. No, it goes before. So what is the connection between olfaction and Parkinson's disease? Can we use olfaction, if not as a predictor, but as a, okay, we need to be testing ourselves maybe more often. We need to have conversations with our doctor if we can smell as well as we did before. I don't know, there's something really, really interesting there. And I'm gonna tell you just a little bit about what happens in the Parkinson's brain. And well, there's a lot of things happening in the Parkinson brain, but I'm just gonna focus on one aspect that will be relevant for our project. In the human brain, we have mostly two centers of neurons that produce dopamine. Dopamine, you have probably heard about it before, even if you're not a neuroscientist. Dopamine is a neurotransmitter that neurons use to communicate. And in the case of humans, dopamine has two major functions. Motor, controls motion, and it also controls motivation and other like emotional behaviors. And we only have two centers in the brain that produce dopamine. These are very, very precious centers. The center that produces the dopamine that we use to control movement is called the substantia nigra, and is found in the base of the brain, okay? So the substantia nigra in an individual without Parkinson's disease will have all of these group of neurons. What I'm showing you here is a synapse, and these synapses are the sites of neuronal communication. So these, because they are dopaminergic neurons, they produce dopamine, and dopamine will send the message. Oh, and I'm sorry, I'm missing people here. So this is the substantia nigra, and these will be the synapses, right? The sites of communication. So in a person with Parkinson's disease, those dopaminergic neurons start dying. So then those synapses become weaker, and the less dopamine there is, the worse the symptoms will be. So Parkinson's disease is called a neurodegenerative disease because it's progressive. So people usually start with noticeable symptoms, of course, and they progressively get worse because these neurons keep dying in the brain. Okay, and they die irreversibly. If they die, again, they don't come back. Do we have any questions so far about any of these? Maybe if I got too technical. No? Okay, I'll move forward then. Okay, so in humans, right, we talked about the olfactory sniffing test. We smell them, we tell the doctor, it smells like this, or I ask you, are you able to smell that? We cannot ask animals if they're smelling. We can't. So how can we test olfaction, and particularly in fish, that's the animal that I work with? Well, we look at their behavioral responses, right? I told you that they respond in very strong ways to different types of chemicals. So for example, this is an example from another project in my lab. We do brain damage. And what you see here is in the top panel, maybe I can indicate. Here in the top panel, we have control fish that have been exposed to alanine. Alanine is an amino acid that is found in food. So when zebrafish smell food, they swim towards that and they swim fast. So we can test whether the animals are swimming towards the source of the chemical. We can test the speed, we can test the distance. So an animal that has received brain trauma like this doesn't smell alanine, doesn't go to that site. It doesn't matter, doesn't make a difference. And in this project in particular, we waited 25 days and our fish recovered their ability to smell. Okay, so we can test olfactory function by studying behavior. But to show behavior, fish need to swim. And problem with Parkinson's disease is that it impairs swimming in fish, right? If we are impairing the ability to swim, how can we test olfaction then? So that's one of the major methodological challenges that we encountered when we were trying to do this project. So we came up with a new approach. And our approach was not to make a model of Parkinson's disease, because Parkinson's disease is widely studied in zebrafish, and we know how it looks like, and we know that these fish lose the ability to swim, etc. But we wanted to make a model, and when I say model, is these animals don't have Parkinson's disease, but we have, we try to replicate one of the things that we find in Parkinson's disease in humans to study that, which is very simplified. So to do that, we specifically wanted to target dopaminergic neurons, but not all over the brain, but only in the olfactory bulbs. So we hoped that targeting dopaminergic cells only there would preserve motor function. And we would have then an animal model that would allow us to test olfactory function and dopaminergic loss with freely swimming fish. Yes, I know. Do you have any questions? No? All right. So how do we do this? So you're gonna see this diagram quite often. Imagine this is the top of zebrafish. Imagine that we remove the skull. We really cannot see the brain like that. That would be very convenient. Sadly, no, we have to kind of, yeah, we know where these parts of the brain are. So this is the top view. So of course, the top is where the mouth is. These two little rounded things, those are the rosettes or the olfactory epithelium there in the nose. And then the first portion of the brain right here is the OB or olfactory bulb. So this is the top of the zebrafish head, okay? This is a diagram. So what we do is that we use an injection of a chemical that we call 6-OHDA. So this chemical is used by other labs, by rat and mice and other zebrafish lab, because it kills dopaminergic neurons very specifically. It doesn't kill any other type of neuron. So it's a really good experimental tool that we can use to kill those neurons very specifically. And the site of injection for us is, maybe I'm gonna go back, in the space here, and I'm gonna show this here so you folks can see it too. In a space here, do you see this little black space? So we have some liquid in there that is flowing all throughout the brain. So we specifically hit here in the middle. So we don't touch the brain at all, we just hit in the middle. And again, and this is just a shout out to my students, these fish are about this long, and yet they find the place where to inject. And we can tell whether an injection is successful or not because we put some color in it so we can track if the color goes all throughout the brain. So, and all the experiments that I'm gonna show you today were done by my undergrad students. I do teach them, but then when they are trained, they do it themselves. So this is, we use a microscope, but it's still pretty fascinating to me, right? That young scientists can do this amazing work. So this is another diagram. This is the side of the brain. This is sort of how it's going to look for the rest of the talk. So in this diagram, we have our injection here in between the olfactory bulb and the back of the brain. And what you see here in this diagram, there's a lot of numbers and a lot of names. I'm not going to spend time on those, but each of those is a center of dopaminergic cells. Humans have how many centers? This is a quiz. 2. Zebrafish has around 15. We don't know why, and this is where it gets tricky, right? They're similar to us, but also different. So we don't know why. But because they have dopaminergic cells in the olfactory bulb, we were like, well, maybe we can target this without targeting the ones in the back. Those are the ones that control movement. This is what we wanted to achieve. And this is the work of these two wonderful students, Nate and Sam. They graduated, but they were the ones who started this project, who injected many fish unsuccessfully. And of course, all of this is approved by our ethical committee. David over there is part of the committee, so thank you so much for your volunteer work. All of this is done under anesthetics and in the most ethical of ways, but it does take a while, right, to come up with a new model of a tiny injection. So both Nate and Sam are the force behind this project. So our experimental approach is the following. We inject with this 6-OHD8 and we follow on 3 different dates. We follow 1 day post-injection, or you're going to see DPI. So that's 1 day after we injected, 3 days after we injected, and 7 days after we injected. And of course, we always have a control group that are animals that have no injection, right? So we can compare the differences. So we do 2 major type of studies. We study the structure of the brain itself and the rosette. So to do that, we take the brains out and we take the rosettes out. Again, really tiny. I can barely see with the naked eyes. Sometimes the brain, like, squint over there. All the dissections are done by my students. Then we put this tissue that is so tiny into wax. We treat it, we put it into wax. And the reason for that is so we can take some slices, like when you go to Meijer and get some ham for your sandwiches, you know, get big chunk, and then you get those little thin slices, it looks like that. It looks like we're just just sectioning bologna, but no, it's actually braids, not bologna, but braids. So in the end, we end up having these glass slides with a lot of brain tissue of zebrafish. And we use this tissue to use markers that are fluorescently labeled. And these markers are the best because they're quite specific. We buy them. They look like this. This is actually from one of the companies. This is a flash drive that I got for free after thousands of dollars spent. Right? These are specific. We get them and we ask the company, well, I want to label dopaminergic cells. Okay, you should use this. Or I want to label neurons. Use that. And then they have fluorescent colors. And why do we use fluorescence? Because we use a microscope that can pick up on those fluorescent signals. So all of the work that you're gonna see today are gonna be pictures with colors, mostly green, red, and blue. And all of these are specific markers for different things, okay? And we also do behavioral studies in which we use fish in tanks of different types. And we add some of these chemicals, different odorants, and then we take videos and track their behavior using specific software that can tell us whether the fish was faster or swam more or stayed in one side of the tank, so we can evaluate then all the behaviors. Okay. Do we have any question about our experimental approach? No? All right. So this is for the results. So what have we learned about in our studies? So in all cases, what I'm going to be showing you is pictures of the brain of zebrafish. This is going to be a side of the brain, and the brain is quite long, quite long. It's like tiny but long. And we look at the olfactory bulb, which is at the very, very tip. So I'm not going to show you all the brain. I'm just going to show you the olfactory bulb, which is very round. So you're going to see these quite a bit, okay? So in this case, we have a marker, and these are the pictures that are published in the paper. This is how they look in the paper, okay? So TH is a marker for dopaminergic neurons. So it will only label that, and it will label that with color green. So whenever you see, this is a magnified view. So I hope that you can appreciate the round cell body, and then there are like little dots around. Those are the synapses. We can know for a fact those are dopaminergic neurons because we bought this marker, okay? This is how a control animal looks like. And right here we do manual counts. We count the cells to know how many there are in a normal control zebrafish. And then 1 day, 3 days, and 7 days post-injection, we see that our injection was actually quite good. It reduced about 50% of the amount of dopaminergic cells in there. So it was quite good. It was quite effective. As you can see here in this panel, there's less green, right? It's less bright because those neurons are gone. And something that I will tell you, we didn't do this in my lab, but we know that these dopaminergic neurons can come back. About 28, 35 days, they start regenerating. So we did not focus on the long-term studies. We wanted to study what happens when they're gone. That's why we focus on 1, 3, and 7 days post-injection. And as you can see here in the graph, by 7 days, there's no increase in cells. It's still too early for that. We also use this experimental group called SHAM. SHAM means injection, but not with the bad chemical. right? So someone could say, well, maybe these neurons are dying because you are poking the brain. So we inject something that is not 6-OHDA, and we see that this injection does not kill these neurons. So then we can tell for a fact that these effects are due to this chemical, and that this chemical is actually targeting these cells that we want to target. And some of this work was done by Sam, which I introduced earlier, and Lexus Putt. They work in these. And when we submitted these for publication, for peer reviewed, we have other scientists evaluating our work and asking questions. And they were like, okay, you're showing us that you're targeting cells in the olfactory bulb, but are you targeting dopaminergic cells in other parts of the brain? Which is a really valid question, right? But we have so many dopaminergic cells. So what we did is that we counted the amount of dopaminergic neurons in other parts of the brain, like in this diagram. I apologize if it's a little tiny, but I wanted to show you the results a little larger. And we found that the only other group that was decreased was this, number 2. And I'm going to show you over there. This is the olfactory bulb, and this is number 2, because of its proximity. But all the other centers were not affected. I'm going to show you here. So this is where we inject. So this is a bulb, and these are the neurons that were affected, but not these ones in the back. So we know that these neurons are not affected, but of course, the million-dollar question, could we actually come up with a model that doesn't have any motor problems? Yes, we did. So what we did for testing this is just putting the fish just to swim. I mean, we control the experiment in many ways, but we just allow fish to swim. And then we test the speed, we test the distance, and we test the pattern of swimming. And we didn't find any changes in swimming. So we know that we target these neurons and that fish can swim. So that's what we wanted. Now, so what happens with synapses? Okay, when we talked about synapses being necessary for neuronal communication, right? I use this example with my students all the time because they basically were born texting people. We didn't, but I'm like, imagine that your text doesn't go through, right? So that's a synaptic, that's a synapse that is miscommunicating. Or imagine that, back in the day when we used to have, maybe some of us still have phones in our homes, like you pick up the phone, incredible. And sometimes you couldn't hear the person well. So that's like a synapse that is not communicating well, okay? And in Parkinson's disease, these dopaminergic synapses are not communicating. So how does that look like in zebrafish? And this is just a reminder of, we have our olfactory epithelium in the nose, and we have our olfactory bulb in the brain. These are the 2 parts that we're studying. And in this diagram over here, these might look like whiskers from, like for beating eggs. It's a diagram. They're not. They're olfactory axons. So in these olfactory axons, we have the sites for olfactory synapses. So if those are messed up, it is likely that fish will not smell well, okay? So we studied how these look like. So what I'm going to show you in the next slide is a diagram that looks like this. I'm very big on diagrams in case you haven't noticed, but that happens when you're a teacher. People don't know what you're talking about, so you need to draw diagrams all the time. And I teach neuroanatomy here at Hope, and it seems like the brain is very hard to learn about. So I'm all for diagrams. So this is a diagram of the olfactory bulb viewed from the side. And we selected 3 of these sites. The name doesn't really matter. What matters to me for telling you is that these are important sites for where these synapses are located, okay? So this is the diagram, and this is how it actually looks in a picture. So here we have a marker that labels specific synapses. Its name is SV2. And you're gonna find it in the picture in green. Then we have TH. Again, TH is the marker for dopaminergic neurons. And then we put them together and we study if these dopaminergic synapses are somehow messed up. Okay, and again, we have 3 sites of different synaptic contacts. So we have our control fish in these 3 sites. I'm going to show you the controls. So after 1 day, some of these look different, but not terribly different. I look at these all the time and I'm like, well, you know, the shape, the whisker shape is not quite there, but it's not awfully lost either. So let's see by 3 days. And by 3 days, I hope that you can appreciate, I'm going to be here and then here, how this looks very different from this. I think I have some colors to show you, so it's probably, there we go. So the shape of these is very different. The colors are like mushed together. In this case also looks different, and this also looks different. So what that tells us is that although these neurons die immediately, because we know that they die, it takes a while for those synapses to be lost or degenerated. And something really, really incredible that we looked at is that some of these synapses come back by 7 days. I know that it's probably a little tiny, but hopefully you can believe me and you can download the paper and be amazed. See it really big. That some of the shape, some of the structure here, here, and here come back. Okay, the structure, the colors, the location of this, like if you compare these two, they look very different. So this is an indication to us that synapses have been restored. And just to remind you, by 7 days, there are no new neurons, right? Neurons have not been restored in any way. However, what we're seeing there is that some of the remaining synapses from other neurons that have not died, we believe that are kind of pitching in and helping cover, you know, the lost friend's job. Like they come and help. This is a process called plasticity. So when we talk about brain plasticity, humans are not too bad at plasticity. We use plasticity all the time when we learn. So as you're learning about Zebrafish olfactory system, your brains are actually becoming plastic. You're learning hopefully a thing or two, and this means that your synapses are changing. So we see this like after damage, these synapses are changing this fast and this tremendously. So we're really happy with that. And this work, all of this work was done by Heather, Grace, and Gabby. Grace and Gabby are in med school, and Heather is in law school, which was very unexpected. It's like, can you write a letter for me? Like, yes, but you know, science and law apparently have a lot in common. I don't know if any of you are lawyers or scientists or physicians, but I learn things from my students every day. So a scientist can also become a lawyer. That's what I learned from Heather. Yes. Do we have any questions so far or anything you would like me to talk about again? Or explain in a different way, maybe? No? We're good. Okay. So yes, we targeted those neurons. Yay. Fish can swim. Also, yay. What happens to olfaction? What happens to the sense of smell? That was basically why we did all of this, right? So I'm gonna tell you about the chemical that we use to test olfaction. So we used a chemical called cadaverine, as in cadaver, and it's basically a rite of passage for every student to smell the cadaverine in the lab. It doesn't smell like the person. To fish, it smells really bad. To humans, thankfully, it doesn't smell bad. Again, right? Like what is an odorant for some species is not an odorant, but it's called cadaver and it's quite grim. So it smells like decaying flesh to fish. So that's going to create an alarm or an aversive response. So in fish, this looks in two different ways. This looks like freezing. Fish are just, swimming, having fun, and then they smell cadaverine and they just literally just like fresh. They don't do anything. They don't move, they just freeze. That's a response that maybe some of us can, relate with, right? Like you're just so startled that you just freeze. And the second response is called darting. With this response, fish start swimming very erratically, very fast. And we have kind of small tanks when we test this. What we believe is that fish are probably trying to escape away, but because they're confined in a small tank, they go and back, they go and back, go and back. So then using our software, we can measure the amount of time spent in the freezing and in the darting conditions. So when we see that there's freezing and darting, that indicates to us that fish can actually smell. Okay, so that's what we are testing. And this is how our testing behavioral chamber looks like. As I told you, it's kind of a smaller tank. And we put the cadaverine in one side and we take video 30 seconds before we put the cadaverine and 30 seconds after. So we can have an idea of how fish were behaving before and after. And we know that cadaverine at 30 seconds kind of stays in the same half of the tank, which is good. So these are just normal swimming responses of controlled fish 1 day and 7 days after injections. So nothing much to see. They're swimming normally. As I showed you before, we did some testing and fish can actually swim well. We did some additional testing with these fish. I'm not going to show you, but we know that fish do not freeze or dart more after the injection. So we can do the study well. So we know that in a controlled fish, when we add cadaverine to the water, fish dart and freeze more. And as you can see in this picture right here, fish tend to go to the bottom, right? Because they think they're not swimming. So when they freeze, they sink. But you see, they sink mostly in the other half, right? Like there's no cadaverine here. So it's an aversive response. They do not want to be near that chemical at all. What happens with our fish that have been injected at 1 day is that they don't care. They don't respond to cadaverine in any way because they cannot smell it. They do not freeze, they do not dart, and they do not avoid the half of the tank that has the cadaverine. They cannot detect it, right? At all. They can't smell it, so they don't respond. By 7 days, we love to see that this olfactory function came back. So again, this is quite unique. Perhaps if we lose olfaction due to a neurodegenerative disease, it might not come back by itself. There might be ways in which we can do olfactory training, but by itself, if we lose olfaction, it's likely not coming back on its own. But zebrafish, they're good at this, right? That's why we study this. So we're like so excited. Yes, does this mean that zebrafish cannot smell anything at all? So then we tried alanine that I talked to you about, alanine, the amino acid. that smells like food. And zebrafish can still smell this. Like we didn't see any reduction in alanine. So this tells us two things, that olfactory function is lost, but only to certain things and not others. And if you think about what chemical is most important for survival, right? It's gonna be food probably. And there are more neurons in the nose that respond to food than neurons that respond to cadaverine. So it's likely that those neurons are the ones that are either dying or losing the ability to pick up on scents. So far we know, again, that some of these olfactory loss is selective. And this happens in humans too. The loss to certain chemicals can go before some other types of chemicals. So this is kind of similar to what we would see in a person. Okay. And lastly, our number 3 question is that what are the factors that drive this ability to recover and function and structure? So you have all probably heard the term neuroinflammation or inflammation in the brain. In humans, for example, if we have traumatic brain injury, or again in Parkinson's or Alzheimer's disease, there's a lot of inflammation in the brain. Chronic inflammation, well, not just in the brain, but in the body, is not good. It's actually quite bad. But guess what? Zebrafish is unique, right? I will say, guess what? Zebrafish is different from us. In zebrafish, 2 things happen. Inflammation is not chronic, it's not there for days and days and weeks and days. It goes back to normal. It does a thing, does repair, comes back to normal. And in zebrafish, neuroinflammation is not bad. It's actually good. So that's where I believe where the juice is for understanding what are some of these factors that help zebrafish heal that we don't have. I think it's in the inflammation piece, and not just me, and there's many scientists that study this. So what I'm gonna show you is, again, pictures of the zebrafish brain. And these pictures now label a particular type of cell that is called an astroglial cell. That is an inflammatory cell that gets activated with injection. So you can see here, there's a little more green in these pictures, and that indicates to us that these cells are becoming activated. And just like any other inflammatory cell, these cells are going to be sending signals and secreting other signals. And we have more results that I didn't bring because I didn't want to go over time. But we know that these cells are recruiting other inflammatory cells also to the brain. So at one day, we see an increase in inflammation. But then I was surprised to find that by 3 days, that inflammation was even further increased. So this wasn't like an automatic thing. This wasn't an increased inflammation. In day number 3, as you can see, more green means more inflammation. And then by 7 days, inflammation started to go down. Again, this is not chronic inflammation that's going to stay high forever. It's going to go down at some point. And to show that these effects of inflammation were specifically due to the chemical that we injected and not to the injection itself, right? Because we're poking the brain, of course, there's going to be inflammation. We co-injected with an anti-inflammatory called franlucast. So we use these 6-OHDA and franlucast together to see if inflammation was down. So this indicates to us that, sorry about that, that this inflammation is quite specific to this chemical. So when we inject, as you remember, our sham group, when we injected by itself, we didn't lose any neurons. So this is an indication, right, that this chemical is doing something really bad. It's killing neurons and it's causing inflammation in the brain. Now, do you remember in my previous results that I showed that there's something happening at 3 days that I was like, something going on at 3 days? Does anyone remember what I talked about? You're like, you have talked so much. I know. Degeneration of synapses. Exactly. So I have a refresher for you because I know I'm just be blah, 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 blah. So this right here, this is my previous result. I'm just bringing it up again. This right here. Just to remind you that by 3 days is when we observe the most degeneration of synapses, right? 3 days. So I was like, this is funny that we see the most degeneration of synapses at 3 days and that we see the most inflammation at 3 days. So then we went back and looked at our pictures, and again, my trusty diagrams are here. So we look at these 3 sites where these synapses are. And I'm showing it to you here in this rectangle. These are the sites where the synapses are. And can you see, it looks almost like a claw, like that. It looks like a whisker, right? That whisker that I showed you before. These are inflammatory cells that are extending these kind of claw-like fingers and surrounding all of these sites where these synapses look bad. And again, I didn't show you this, but we also know that there are other immune cells that are coming to the site to eat some of these debris away, and then they leave. So we believe that these neuroinflammatory responses are helping in that recovery of synapses. They're taking some of this debris away. They're taking all of those synapses that are lost and broken and we don't need, so the new synapses can be formed. Again, I wish we could do that. We're not as good as Zebrafish as doing that. And lastly, I have been focusing a lot on the brain, right, on the olfactory bulb. But as I told you, we have 2 components to olfaction. We have the nose, that is the one that is getting those chemicals in the first place. And then we have the brain. So yet again, another diagram. You have seen this before, so you obviously are experts by right now. You've seen this 1,000 times. So we have our rosette here. And although olfactory information flows from nose to brain, right? From the nose, picks up the odors, gets sent to the brain. The communication between these two is bidirectional. They're best friends, right? And we know we don't want to be the friend who's always calling the other person and never getting a response. Like, I'm inviting you to coffee, you're gonna invite me back at some point, right? It's bidirectional, like a friendship. So what happens when there's loss of neurons in the bulb? What happens to the nose? If this is a bidirectional communication, if this is a friendship, what happens when one of the friends is no longer responding, right? So this led us to study the olfactory epithelium, the nose, like is there anything happening? And just to be very clear, we do not touch the nose at all. We do not inject anything in there, nothing. We do nothing to it, right? So this is how a rosette looks like. It's really, really beautiful shape. And what you see in green are actually neurons. They stay in the center of the rosette, and that is the sensory part because that's the part that has the neurons that respond to smells. So we saw that after injection, we see a thinning of that neuronal layer, as you can see here. It's less bright, it's a little thinner. That indicates to us that these neurons are either dying or that they're not functioning properly. But guess what happens by 7 days? They're back, baby. You know it. They're back by 7 days. So indeed, when the friend suffers, the other friend responds also by degenerating. But by 7 days, it's back to business. So we wondered, how does this happen? How can we study these regenerative properties? So we do this by doing a new experiment that I'm going to tell you about really briefly. So after we inject the brain with our chemical 6-OHD8, right after that, we don't do this simultaneously. We wait and then do the other, but for diagram purposes, we'll flip the fish and inject a chemical called BRDU. This BRDU chemical is going to stay in the system for about a day or so, and it's going to label every cell that was newly produced what the BRDU was there. So this means that a cell that has a BRDU flag is a new cell. We know it. There's no other way that a cell has BRDU unless it divided when we had the system. So it's a really, really good system for tracking new cells. And just a quick reminder, we just talked about this. In the rosette, we have the sensory and the non-sensory parts, right? Both are important, but the ones that are important for olfaction is the one in the middle where the neurons are. And the reason why I'm emphasizing this is because we wanted to test, are there any new neurons and new cells in the epithelium? And to know that, we can tell whether a new cell is a neuron or not a neuron based on where it is. If it's in the middle, in the sensory part, it's a neuron. If it's outside, then it's not a neuron, okay? So we use this BRDU label. Oh, I don't think you can see this very well. I apologize, but from my end, it's beautiful. You have to believe me. This is a control. Can you see more? Yeah. So we have a lot of new cells, but I hope that you can appreciate in the top image maybe that most of these bright dots are around, right? So these are all around. By 3 days, we have even a little bit more. And by 7 days, some of these cells are already in the middle. They move. And they move when they become neurons. They are born, they decide, I'm gonna be a neuron, I'm gonna help out, I'm gonna move where neurons are. That's how this happens. This is a magnified view of all of those, where you can find that at first, most of these bright BRDU dots are outside, but as time goes on, these cells start to move in when they become neurons. So these results indicate that in the nose, by 7 days, there are new neurons in the nose that are helping these tissue regenerate. And this is the last of my, this is the last of the results that I'm going to present to you. But in the future, what we want to know is whether these new neurons in the nose are making those new synaptic connections that we see by 7 days. That's our hypothesis. So finally, I hope in nothing else that you leave today with the idea that zebrafish are cool, that my students are even cooler and capable, and that we can actually use zebrafish to study human disease. And we're a little bit short on time, maybe, so I'm not going to read all of these to you, but this is what we believe is happening, right? We believe that the system is able to bounce back really quickly by 7 days. We believe that some of these neurons in the nose can connect And we know, not from my work, but from the work of others, that dopaminergic neurons in the brain can actually be also reborn and come to the olfactory bulb. So it would be an interesting project for us to see if these two neurons can connect and form friendships, and if this new friendship is what's allowing fish to recover olfaction. And these are multiple people who have worked in my lab. I like to tell to people that I have a small army of students. Like, all of these students have gone through my lab in only 5 years. Some of their faces, and very, very talented people. I like to, of course, acknowledge them, acknowledge Luis for inviting me here today, and my funding. This research could not be done without funding agencies. In particular, this project was funded by a private foundation, the Kenneth Campbell Foundation, which is very unknown. It is based in Grand Rapids, and I believe Kenneth Campbell had Parkinson's disease or any other sort of neurological disease, and his wife manages some of these beautiful gifts to investigators that allows us to do all these beautiful research. So I'm open to questions if anyone has any.
David, are there any questions in cyberspace? Okay. Any questions in the room? I have a couple.
Yeah.
Going back to the early part of your presentation, most people here are well aware of taste buds and the fact that we have receptors in them specific for salt and sweetness and bitterness.
Yeah.
Tell us more about the receptors that we have to be able to detect or discriminate 1 trillion or more compounds.
Wow, that's a very good question. So I'm gonna go. So in the tongue, we have basically 5 types, right? So as long as there's a sugar, that sugar binds to the cell, and we interpret it as a sugar. In the olfactory system, it's way more complicated because 2 molecules can look almost the same, and one of them might smell like chocolate and the other one might smell like diaper, like completely different. So we cannot predict how a molecule will smell based on their chemical structure. That's one thing. Another thing is that, generally speaking, zebrafish and humans, we have 1 receptor per type of molecule. So for example, all amino acids will bind to the same type of receptor, but each type of receptor might have 100 different copies with different variations. That's another complexity. Another complexity is that, for example, the smell of a rose. There's not like the smell of a rose. It's probably a dozen chemicals that smell of rose. And they're different roses, right? And sometimes if you're good, you can distinguish between them. So it's a matter of combining aromas that have dozens, hundreds of different chemicals. Do you enjoy drinking wine? Wine, yes?
Oh, yes.
Oh, yes, absolutely. So when we say that wine smells like molasses, right? It doesn't have molasses in it, but it has chemicals that molasses also have, or ripe berry fruit, or there's some wild things like cement. Like, we've never tasted cement, right? But that like mineral-ish quality that we use. So in short, it's very complicated. We cannot predict how a smell will smell like. And there's people who are trying to use artificial intelligence to solve this problem, but it's very, very complicated.
And one final question. So many people were affected by the SARS-CoV-2 virus. Can you give us a glimpse of how that virus affects the olfactory system? Is it a specific cluster of nerves? How does it impair the olfactory system?
Okay, that's another fantastic question. So I'm gonna go back maybe. And zebrafish is used for studying that, which is not surprising. We know, amazing. So let me show you a little bit of here. Okay, so in the olfactory epithelium, we have all of these colored little things that are neurons. But we have other types of cells that are called sustentacular cells. So these are not neurons, but they exist there to give sustenance to the neurons, to give structure, to help out. Those are the cells that get infected with SARS-CoV, with COVID-19. Not the neurons, but those. So they get infected. There's a lot of inflammation. Again, inflammation in humans is bad news. And sometimes the virus can enter the brain through the olfactory nerve. Something I didn't tell you is that in between the bulb and the epithelium, there's bone, there's the skull, right? So the skull that protects the brain against all of these different viruses, there are little holes in there so those axons can go through. So there's a lot of viral infections that can enter through the nose. Even drugs of abuse that are snorted go to the brain super fast because of that. So SARS-CoV can enter the brain through the olfactory nerve. Yeah, but this was very surprising that it does not infect neurons, but it infects other cells. We don't know why some people recovered their sense of smell and some didn't. Some people didn't lose olfaction at all. Some people recovered olfaction, but in a different way, like certain smells were weird afterwards, or vinegary, or like usually bad. So we don't understand that fully yet.
I have a question over here.
Yeah.
More of a comment. In 1956, I was a 10-year-old here in Holland, Michigan. And all of a sudden, the air smelled different, kind of metallic, and I couldn't figure it out. Tornado was coming.
Metallic? Wow. Well, yeah, we can smell when it's gonna rain, right? It's ozone. It's like this oxygen, a different type of oxygen, and we can detect that. It's like we can, of course, smell the rain, but we can smell when it's going to rain. How incredible is that? And there's a line of thought that, like, we know our companion animals like dogs and cats can detect some of these cues before raining, when we're approaching, even before earthquakes, that some of them have other ways of perceiving things, but there's a line of thought that they can smell things that we can't. It might be that, like, yeah, for sure. There's things changing in the atmosphere, in the air. So yeah, I wouldn't be surprised. What type of metal, if you remember? It's just like a car, or like rusty?
Just metallic.
Okay. Yeah. Good to know. If we smell metallic, immediate shelter, for sure.
One
Yeah.
last one.
Does any of this affect our longevity?
What specifically?
How much we smell, or a loss of smell? Like, can it predict your lifespan?
I know that individuals with when I say olfactory loss, there's a degree, right? There might be people who cannot smell very well, and there might be people who can't smell anything. So there are risks associated with not smelling that are safety risk, like eating something that is spoiled or not detecting gas or things like that. So people with severe olfactory loss as a group might have a reduced, like a shorter lifespan. Not necessarily that a single person will live less, but as a group. I do not know if having a good sense of smell would increase longevity. But it's also known that people with with olfactory dysfunction sometimes present depression and other anxiety and things that are related to their inability to pick up on cues that we're not aware of, like social cues, like smelling your partner. Maybe you can't smell them anymore, right? We're talking about these information that we are getting all of the time that we're not even processing, like not just visual, but this person who did the study on the chocolate sniffing, they have another very interesting study on handshaking. They record people in an office setting. So then people shake hands, and they counted the times that people who shook another person's hand brought the hand to the face 3 times more. We're smelling people, right? We don't do it on purpose, but we have this tendency to get these chemical cues from others. I thought during the pandemic how interesting it was that we couldn't smell each other because we were masked. We could only smell ourselves. I was like, this is something that they probably will study, right? Are we missing important communication from other people? So yeah, it might be that olfactory loss may decrease quality and maybe expectancy. The other way, I don't know, but that's a very interesting question. I never thought about that. Thank you.
Please join me in thanking our technical producer, David Blatt, and our speaker, Dr. Erika Calvo-Ochoa. Thank you so much.